that example given in the video clip. If the mother takes care of the baby very well, that habit is transferred to the mother. Because the baby is from that mother's family, still, if the baby got into adopted good mother, and that character can be transferred. This is typically one of use and disuse the rule. So totally this is not genetics. Something strange thing was observed. And funny thing, father's role is not that big. Father really don't need babies to go to mother. Okay? And the gene does not bother with that either. Family is also engine, does not take much attention. Very strange thing is going on. Okay, C methylation. Out of ATCG, U methylation, T. One of this is in RNA. Which one is in RNA? This is in RNA. It's a partner in DNA. It's a T, but missing. G is missing. So C methylation is the molecular marker of the genetics. C methylation, when you say C methylation, it's a DNA methylation. RNA also has a C, but we are not talking about RNA methylation. And RNA doesn't matter, histone has its own role, but C methylation is on DNA. Okay, very good. Even though C is methylated, still DNA replication is okay. DNA can be replicated because phosphine and quick pairing is still not bounded. Methylation happens on the back. And euchromatin means higher expression. It's open DNA. What is the opposite of euchromatin? Heterochromatin. It's more aggregated form. It's not open. So DNA methylated, then DNA forms more aggregation. Heterochromatin is the form. Even C is methylated, still DNA replication can occur. And DNA double helix can form. Can make the partner of what's next DNA. Only the effect is for mRNA synthesis. mRNA polymerization is a decrease. Histone also can be modified. Histone is a DNA binding protein. It has methylation, acetylation, ubiquitination, different kinds of modification. Methylation effect sometimes increase, sometimes decrease. It's not consistent. So that's case by case. Should be on the red. And the next one is asking acetylation. effect, it can increase euchromatin, increase mRNA expression, can be explained like this. Histone is a protein, and DNA is binding to this histone like this. DNA is charged positively, negatively. Negatively. That's why if the histone is more positively charged, interaction may be even more compact. Right? If this the lysine motif is methylated and charge changes, charge is not changing. It's still positively charged. But if this is acetylated, how about it? Charge is removed. It becomes neutral. Okay? Acetylation reduces histone charges. Then interaction with the DNA will be decreased. Right? So that's why DNA becomes more loosened and it's more open, form euchromatin and make more mRNA. Makes sense, right? Methylation does not change the charges. Even actually, if you have a more methyl group here, the acidity of this amine is increased. That means positive charge is stronger. Okay? But that does not mean that methylation on histone decreases the mRNA expression. It is kind of sometimes increases, sometimes decreases. Only clear effect is on acetylation. Acetylation increases gene expression. Okay? A really tricky question. This methylation pattern, whether majority of them can be transferred to the upper springs? No. Very small amount of methylation is transferred to the upper springs. Very small amount. And there's some examples for that. Majority of them is erased. Maybe 99% of methylation is erased. That's what the Lamarck's theory was wrong. They tried to make a longer and longer neck, but that character cannot be transferred. So majority of it disappeared, but some small amount of methylation survived and transferred. That's epigenetic. When we say genes order, if this is upstream, then this is what end? 5' end. This is a 3' end. So maybe this is the gene area, and for one gene, they need to have some upstream sequence and downstream sequence. And upstream sequence mainly CPG is located here. So methylation make this gene downregulated. Demethylation opens this up and upregulated. Okay, so this is called as upstream. This is the downstream. And time to time there is some intron part inside here. Some of those introns they have a functional role as ribosome. They have its own functional role, and create this and then reconnect with splicing can be catalyzed by the intron structures. So there is some example of RNA word, like a functional RNA, like this. Okay. That's the question. This is 5', this is 3'. This one, that's a GPC, right? That's a different. And here, no, again, GPC and CPC, only one here. Okay? Order is important. CG, you count. GC, you don't count. Then, percent-wise, CG out of total populations, how many percent by average? That's kind of average, but if you look at those maps, that area is highly condensed on upstream of genes, not randomly distributed. So that means it's evolved to make those regulating the regions are concentrated on probability. Okay, so this is seems like you guys are following study quite well. And today I invited June volunteered to give this presentation today. Okay, please welcome June.
Hello, my name is Han Ji-hong, and today I'm going to introduce about the unique cases of epigenetics. These are the contents. And last lesson we learned about the Darwin's evolution theory called natural selections. And according to this theory, the species or individuals which has fittest phenotypes or genotypes have a highest percentage to survival, and this process begins for a long time, and this turns into evolution. And this theory is considered as orthodox theory due to as time goes by and other biological discoveries were shown, like Mendelian evolution, or the genetic material is discovered as DNA or something. And according to this theory, Lamarck's use and disuse theory is some kind of it is not a good theory, it seems. But however, Darwin's theory has critical limitations. It's shown as this. We can make these kind of questions. So, is genetic materials only determines the phenotype change, or what about the same genotype and different phenotype cases? In these cases, we can also see the differences, even though the individuals which have similar genotype shows significant phenotype changes, differences. So I'll show these kind of several cases in the next page. The first case is Dutch hunger winter of World War II. During the 1944 to 45, at the end of the World War II, after the Normandy operation was complete or something, and in this season, the German army prevented supplies coming to the Netherlands so that on the operation. So the Netherlands people will suffer from the starvation or malnutrition due to this operation, and these are the pictures. And on this seasonal graph, you can see the famous season, which was not supplied on the season. So on this period, this incident greatly influenced on the mother and their children's phenotype. So as you shown in these three group, late, mid, early, the late groups were the mothers who experienced malnutrition at the end of the fetus development season, and the mid is in the mid-season, and early is the group who experienced malnutrition at the beginning of the fetus So, but among these three groups, the early group and late group showed big difference on the baby's development and growth. Early groups usually have babies which have average weight. It tends to, but as they grow up after the war, they have more chance to be overweight or obese or have diseases like high blood pressure or diabetes kind of something. And the second generation people usually tends to have offspring who has more weight than the normal children. On the other hand, the late group have a baby who has lower weight than the average. And the second generation group tends to have lower weight compared to the other people. And these kind of phenotypes came to normal stage after the third generation. So this case shows that on which fetus development stages does the mother experience malnutrition. This malnutrition season can affect on ventilation pattern on their children. And the second case is fur color of agouti mouse. Agouti mouse usually have this large A gene, and the hair color as shown here. The hair color usually defined by this A gene, and it shows black and yellow mixed patterns. However, if there is some kind of mutations on this gene, the small a or AVY agouti viable genes are shown when the mutations were happening. So researchers did some experiments on agouti mouse. First is when the researcher grow the wild type mouse, they usually show large A type gene. So the most mouse shows this model type hair or some kind of this hair usually. However, when the retrotransposon DNA was inserted in the agouti mouse, these RTPS transcribes RNA, not the DNA, not the protein. And these RNAs transcripted from RTPS deranges the agouti gene regulation patterns, so agouti gene was overexpressed and the AVY gene was usually expressed, and agouti mouse shows yellow hair usually. However, among these AVY gene mouses, when the RTPS on which weight the methylated retrotransposon was methylated, the mouse pattern shows different. So even though mouse has AVY gene, on which rate the RTPS was methylated, they shows different colors of hairs. So for example, if RTPS was not methylated, they usually show yellow patterns. But however, if RTPS is highly methylated, they usually show pseudo-agouti colored hair. And the final example is calico cat. I think someone might know that normal calico cat is almost every normal calico cat is yellow. Every normal calico cat is female. And this is because of the cat's genetical system and X inactivation epigenetical mechanisms. Cat's hair color is highly dependent on it depends on the melanin pigment, which is inside the melanocyte cell. This melanocyte cell is differentiated from the stem cell, and they kind of cluster or kind of group. So which means that look at these cats. These cats have orange or black or white hairs. So the white hair range shares the same stem cell, and the orange skin shares the same stem cell. In this situation, the hair color gene is derivated from the X chromosome. Like, yeah. So in this case, the male cat does not have various colors because they get X gene only from mothers, not the fathers. However, in case of female cat, they get paternal X and maternal X from their mother and father. So in this case, they experience random X activation, and so maternal X can be expressed or paternal X can be expressed. So in this, which X chromosome is activated, their color is different, and this also affects on the melanocyte stem cell and melanocyte cell. So as a result, this cat shows more than 3 colors in their fur. Yes, sir. Okay, this is my presentation. So thank you, and I would like to make questions.
Any question to you?
So male cat has Klinefelter syndrome, and then the cat has
Klinefelter, which means that if cat has more than 2 X chromosome cases, I think it might be possible. So similarly, if female cat has Turner syndrome inside their gene, I think that cat might have only 1 colors.
Okay, we will contact you later. Thank you. Okay, to introduce several cases of some examples of the epigenetics. I also put up those questions to on the PLMS, and then you suggested several other examples too. So he already mentioned that the Audrey happened, so that very charming, so even though eating don't get fat, kind of gifted the shape of this phenotype is actually derived from that famine season of the Dutch of the winter. So in Jung's example, there was 2 cases, right? So when the baby was growing, early stage hunger versus a late stage hunger. Which case is more Darwinian principle phenomenon? Early case and late case. Did you forget? Let me show the genes again. So when the baby did not get enough supply, when early stage of the pregnancy, let's just say human pregnancy is maybe 10 months, and maybe 3 months time, and maybe 8 months time, and they didn't get enough nutrients. And then, which case showing that is Darwinian, the principle is correct. If you don't get enough nutrients, they are born as small. That's natural. But the next generation is normal. This is more Darwinian. If you don't get enough food, then you don't grow well enough. So we become very small and thin. But that character does not transfer to the next other generation. That's Darwinian, right? Even though I don't get enough food for now, my gene is not changing. My gene is transferred the same to the offspring, then it should generate regular, original, my father's the shape should be transferred. Right? But very interestingly, very early stage, 3 months the pregnancy phase, if the baby does not get enough nutrients, very interestingly, when it's born, looks like normal. When it was early stage, it did not get nutrients enough, but the later stage, it gets enough nutrients, 7, 8 months, then the size becomes normal. But when it's growing up, very funny, this baby has more chances for the diabetic and fatty later in 20s. And even this character is transferred to even the next of the generation. Something wrong, right? And when you check the DNA sequence, it's the same. DNA information is the same. Genotype is not changing. But this kind of thing is happening. This is very shocking the observation. Somehow the genotype is not changed at all. That effect is transferred to the next generation. And Odin-Hepburn was one of those cases. And then Odin-Hepburn case is the later case, the case. She looked like the thinner. And during this time, it was not designed, but many people passed through the same the situations for over 1 year of time, and they could be monitored for the next generations. Next 10, 20 years of the time, how they develop into what kind of thing. So it seems like those the problem happened quite early stage here. This early stage effect can remain for a long time, and it can be also transferred to the next strings. Some of the examples you guys have got is as an example of epigenetics. Folic acid, okay, when the flower, the plant, the flower, the flower, they need to experience some cold time. Then it can go into the flower. That can explain that some gene is suppressing flowering. And during those cold treatment, this gene is suppressed by epigenetic modification, means methylation. If the sequence is not changing, but that gene is methylated when the plant goes into the cold treatment, then the gene is turned off, and flower starts. When the ladies get pregnant, usually they hear to take a lot of folic acid. Folic acid is to maintain DNA structure by changing the methylating pattern and make the gene more stable. Means more methylated or less methylated. My question was, DNA is more stable when it is more methylated or less methylated? More methylated, more aggregated, and less chance to be attacked. If gene is become loose and then open, then it is vulnerable to be attacked by many different attacks. So we are talking about right now very early stage of development. This is related to aging too. Okay? So folic acid helps to maintain those methylation and helps keeping those the stable status of the DNA and prevent any kind of malformation of the babies. So this is another example of epigenetic modulation. So the question is, for a long time, here the nature versus nurture. Nurture means giving nutrients. Right? So nurture means the environment. Nature means our genomic sequence. Whether our gene is important or environment is important. Seems like most of them, but seems like the nurture is quite important too. And the question is how? How this kind of environmental factor can change us, also change the genetic information to the next strings, the next of the strings. So genetics is defined by gene sequence. And if gene sequence is the same, then phenotype should be also same. But somehow there is a little different phenotype. And in this case we call this epigenetics. Epi means it's above we are looking. Gene is not changing. So let's say gene is like the information recorded. So this information is exactly the same. But who plays that and who is the director? Now the effect can be different. That means which part of the gene can be more stressed than the other, and which part of the gene can be temporarily suppressed or activated. That's the epigenetics. So this example I gave you in the video clip. So there is a good mother and bad mother. Bad mother's babies are become bad mother. Good mother's babies, they become good mother it can be interpreted that they may have good genes in them. But if they change their babies, still, good mother's baby, whatever the genetic background, they become good mother. At least one of them seems like we found that by the DNA methylation. So that's why, until last week, the classical evolution, we said Darwin was correct. If the gene is the same, or the information is stored in the gene, DNA is transferred, and that dominates those rules. That was the dynamism. And at the moment we said Lamarck's the hypothesis is wrong, but it seems like there's a new other phenomena here, and then there is something we couldn't explain so far, but there is something ongoing like that. So far, phenotype is what we can see, and genotype change means it's not breaking DNA; it's just changing the sequence of DNA. Okay? DNA is damaged, and that does not mean that DNA is destroyed. DNA is the sequence which changes, so-called mutation happens. So if this is matching, that is what we call the original, the Darwinian or the Mendelian of the genetics. Now, when you look at those DNA structures like this is a DNA and this is RNA. Right? How do you know? So hydroxyl group here, next to the hydroxyl group, this is ribose, that's the RNase ribose. This is deoxy, so that's the DNA D, deoxy. Okay. Now, here in DNA, they use A, T, C, G. Here only U is a change. When they make the Watson and Crick pairing, they use this side and they use this side. So they match, this one match to here, this one match to here. And if you look at those methylation at the side of these two, T methylation, T here, T makes the decoupling this side, and this is behind, on the back. Okay, this is the base pair inside. And uridine does not have the methyl, T has a methyl. So even though they have slightly methylation difference, but RNA, DNA, they can make the double helical structure, means this method, the methyl group does not bother for forming the Watson and Crick base pairing. So this one is somehow, this one is chosen for the RNA and DNA. Now if you look at C, and methylated C, if you look at, methylation happens here. That relationship is very similar to U and T and C and C methylations. The effect should be almost similar, but somehow nature chose this as a signaling. That means the partner of recognizing of this site, not only C, maybe CPG together. CPG site is methylated, and that part can be recognized by different enzymes, and they recognize this as a switch off signal. Interestingly, this one was not recognized that way. Structure-wise very similar, but nature shows this C as signaling part. So that methylation is now at least one answer we know now. And that one, if that position is methylated, then that position is somehow suppressed with gene expression. That's what we know. How many methylation sites here? Not sure? Okay, let's count. So here's one. We don't count this. We don't count this. Two. Yeah, two. So one here and one here. And if you make the complementary sequence coming here, then that's also CPG, it makes C and G, this direction, right? That means methylation happens on one side, then the other side is also methylating. Okay. Now here, as I said, let's say this is the gene, this is the upstream 5' directions, this is the promoter area, and the majority of those CPG comes on their positions. And the effect of this DNA methylation is affecting mRNA synthesis. mRNA synthesis is called as a transcription, right? That's why this is a transcriptional silencing or gene knockdown. Gene expression is down. And this effect, in the quiz, we said methylation pattern or epigenetic effect, very small amount is transferred to the next generation. Methylation pattern, when sperm is formed and egg is formed, still this is methylation of the degree. Sperms, they carry methylation pattern. Eggs, they carry methylation pattern. They meet together, then this the fermented, the fertilized egg cells should have an average of these two, about 50%, let's say. Now, when they developed into embryo, 99% of methylation is deleted. Very small amount of methylation remained here. And how and why, that's also the topic of the study. But majority of those methylation is removed. And then they restart to give those methylation pattern all the time. That means when those cells forms in the beginning, up to here, and then formed so-called stem cell, which is the topic of the next week. When they formed these stem cells, those genes has almost no methylation or lowest methylation, means majority of the genes are open form. And almost every genes are making their proteins. And then when they develop, now those methylation is increased. That means different part of the body, their expression of the genes is also decreased. That means, most likely, in the beginning, those stem cells or the original cells, they should do everything. But when they develop into different part of the body, now some of them becomes the blood cells and muscles and liver cells, different part of the cell, then they should have specific functions. Then the way nature shows is they have everything in the beginning. Now they delete some functions, function by function. And yellow color is open one. Green color, the blue color is a closed one. Originally, they opened almost everything yellow. This is genes, 25,000 genes. Now, depending on their the functional organs, they close some function by function. And they become the specialized liver cells, specialized muscle cells, like that. That means, originally we have 25,000 genes. Then one type of specific cell, let's say muscle cell, we can assume that by looking at this, we can assume that maybe less than 10,000 genes are expressed. If you look at these numbers. Some other cells, they have a little more expressed, but still, it's like about the half, about the 12,000 genes, maybe. So that, how do they control that? If you think, if you needed to turn off gene by gene, then maybe too much work. That's why maybe those DNAs are this kind of big open structure in the beginning. This is euchromatin structure, open structure, express almost everything. When they develop into different cell types, most likely they close chunk by chunk, this area and this area, chunk by chunk they aggregate and close it. Then the end result of this will be shown like this. Big part here, these guys are all closed together. So mechanically this may be easier way. Then to do this, they need to test the many times and then try to connect, relate the genes into one area. So those genes, maybe 200 genes here, this is the liver related gene, and they should be open only in liver cells. All other cells, they should be closed. So that's the result we see here in DNA, the pictures. White picture is a eukaryotic user. Black picture is a heterochromatin user. And seems like they attach to the nuclear membrane, and then around that area, they aggregate to those DNAs. So this kind of black color will increase of the development. But different area will be closed for different types of cells. Okay. That's what is going on. Seems like this is development for each organ level. They develop into different directions. Now that's why, if you look at those, the different organs, cells, and take out and then analyze those genes, and we just said this kind of pattern is defined by methylation on C. So that's why different position of the DNA C methylation pattern is all different depending on different the organs. Now, and the result is depending on this methylation pattern, they make a different mRNAs. Right? They make a different mRNA. Let's just say one cell makes a 10,000 mRNA. You take those mRNA and reverse transcribe it. Then what do you get? That DNA is what you call cDNA, complementary DNA, which is a template to make this mRNA. Right? Then each organs, they will have a different cDNA library. Okay? So that's why if you hear cDNA library, then you should ask, what species? Is that human or mouse? What organ? What age? Depending on the age, they also will change it. So this is 2-month-old mouse liver cDNA. Then using that DNA information, if you express that into mRNA, you can make protein sets for that specific organs. Correct? If you want to make almost every human protein, what should you use? Using one cDNA, and you wanted to make almost every human protein, what cells should you use? You should use a stem cell. Stem cell has expressing almost everything. Then you collect those mRNA and then make a cDNA. That covers maybe the universe, the human protein cells. Okay, so that's why the collection of all the genes, collection of all the genes, what do you call? Genome. One transcript, one mRNA we call transcript, and if you connect them together, we call them transcriptome. All means is collection of everything. Genome, transcriptome. Protein, protein. So that means the collection of every protein is a protein. But depending on the different tissues, proteome may be also different, right? Different tissues will make a different transcriptome and eventually make a different protein. That's something you should keep in mind when you do design you are expecting. Okay, so after transcriptome is the mRNA sequence. Now we need to know this transcriptome is maybe related to the epigenome, because if it is methylated, then it will not make the mRNA. Right? Then transcriptome may be similar to non-methylated genes collections. Make sense? If the DNA is methylated, then it will not make transcriptome. Right? That's why epigenome sequence is matching to non-methylated area is almost similar to transcriptome area. Okay, so this is on DNA level. DNA level, methylation is driving force. Methylation site is aggregated with heterochromatin and suppress those mRNA expression. That effect also coming from DNA binding protein, histone. Histone is a protein. Protein has 20 different amino acids. And then some of those amino acids can be modified. The biggest modification site is a lysine. Lysine has this amine group and arginine. This is arginine. Lysine, arginine, they can be either methylated, acetylated, and phosphorylated, ubiquitinated, by different kinds of post-translational modification can happen to the position. And open form, again, euchromatin, and the closed form is heterochromatin. And depending on those patterns, other effect, methylation or phosphorylation effect, sometimes up and sometimes down. But only clear part is acetylation. More acetylation is open. More acetylation is open, then they start to remove those DNA methylation category. So they are connected. The more often methyl group is removed, it's further open. If it's methylated, it starts to collect these the deacetylates, remove acetyl group, and it's a closed like this. So both of them is working together. DNA methylation pattern and histone acetylation work together. So there is two enzymes. One is acetyl group transferase. Then add more acetyl group to this is histone. This is the green one is histone. Add more acetyl group, then it opens further. And this is euchromatin. Deacetylase work here, then remove acetyl group. Then it will close, it will make beta-glucanate. And in this case, histone acetyl transferase is add this, and then removal. This one is histone deacetylase. This is what we call HDAC. HDAC, HDAC. HDAC is removing acetylase. HAC is acetyl adding enzymes. So let me ask you, if you have more HAC, what happened to the gene expression? Gene expression increase. HDAC, if you have more HDAC, gene expression suppress. Now, some of the cases in tumor cells, tumor treatment, cancer drug, one of those cancer drugs is HDAC inhibitor, is used for cancer drug. Stem cell is more often make more proteins. In common cases, cancer cell also more aggressively growing. It's more similar to stem cell rather than differentiated cell. They have a more open gene structure. They make more proteins. If you keep that in mind, then first of all, DNA methylation versus DNA demethylation, which one would you take to kill the cancer? DNA methylation, right? Then cancer cannot make more proteins. For the histone acetylation, deacetylation, would you like deacetylate or acetylate? Deacetylate. But very funny thing is, HDAC, deacetylation inhibitor, is used for cancer drug. How will you explain?
Because tumor suppress.
1 second. Let's take just a half a minute to think. HDAC inhibitor is being used for cancer drug. It's a little opposite to common sense. Gee.
Tumor cell uses HDAC as kind of tools to edit the body mechanisms for repressing tumor cells. So by using HDAC, tumor cells can grow because HDAC suppresses human tumor suppressor.
Okay, very generally speaking, stem cells have many open genes and make many proteins. True. Cancer cells also open many genes and make a protein. That's also true. But those sets of protein may not be exactly the same. Right? For the tumor cells, there is a tumor promoting the gene, so-called oncogene, or tumor suppressor gene, and tumor is successfully developed. That means that somehow they suppress tumor suppressor gene, they overexpress the oncogene. You can assume so, right? It's already tumor. That means for their own favor, they suppress the tumor suppressor genes. They increase oncogenes. Oncogene make very fast growing. Tumor suppressor gene is checked very carefully every step. If anything is wrong, then kill the cells by apoptosis. So somehow the balance is moved to the tumor's favor. So in this case, any treatment, HDAC inhibitor or even HAT inhibitor, in any cases, if you disturb it, somehow maybe, in this case, the explanation is maybe tumor suppressor gene was highly methylated in tumor, it's suppressed. If you add HDAC inhibitor, it can suppress other genes, open up other genes too, but maybe this gene is more activate for the tumor suppressor gene, and at least as a result, it seems like this effect is more favorable to cure cancer. That's explanation. But that means we don't know which direction is going to work as an anti-tumor. Anyway, all we know is HDAC inhibitor open up the gene, and somehow it increase the gene expression. That's all we know. Which part it affects, we don't know. But some trial other cases seems like HDL inhibitor is effective as a cancer treatment. Okay, some of those your. Okay, so this is summary for the histone modification. Acetylation is clear. Adding acetyl group, activate, euchromatin, increase the gene expression. Deacetylation is out. For others, it's a mixture effect. Methylation or others mixed effect. Okay, one of those examples that you gave also that we heard about the X inactivation. Ladies have 2 X chromosomes. Male has only 1 X chromosome like this size and this size of the Y chromosome. So that's why if 2 X chromosome is all expressed, then the power of these 2 genes expression may be too powerful. Y chromosome is a fragment of X chromosome. It's usually has a matching partner here. They control each other. So try to control each other. But if it's 2 copies, maybe it's a balance is too much. Maybe the difference between man and woman may be much bigger than now. Okay, we are already different enough, right? We can understand the woman, right? We are already very different, but if we have totally 2 different sets, that difference may be too much. So that's why the nature chose. Females, they have 2 copies of X chromosome. They choose only one of them to use. The other one closed down. Mechanism by semen station. That's epigenetic modification. That's why this case is epigenetic modification. Seems like all of the body, the females has all of the body, every cell has 2 copies of X chromosome. And which X chromosome is deactivated? Seems like so far it looks like a random. And area by area. So let's say, as Ji-Young said, maybe 3 copies of X chromosome there. Then from 1 father and 1 mother, there's 3 kinds of X chromosome. 1 from father, 2 from mother. Okay, and then it goes into the distributed to the babies. And for male, it has a chance only to take only 1 of them. Right? Then there's no chance to have mixed colors. Females, they have at least 3 colors can be mixed, and the end result is seems like, and now we can see we can know which is male and which is female, right? Most likely this is male, only 1 color. What color? It depends on which X chromosome it takes, white, black, or yellow. Right? So it seems like this one has yellow and black, 2 X chromosome, and then randomly expressed that this area is black means this area is yellow means kind of situations. Maybe a little more complicated with the 3 color. But anyway, in general, seems like this is already should be females, and then this single color that should be males. We have another case similar to this, colorblind. Right? Colorblind case, male is more vulnerable for the colorblind because that gene is on X chromosome, and if you have 1 active, then you are okay. Both of them are inactive, then you are colorblind. Male has only 1 X chromosome. But if that has a problem, then no choice. But female has another one is healthy, then they have a better chance to survive from the colorblind. But that case, there is a on and off, right? But in this case, 2 X chromosomes is involved here. 1 should be inactivated. That's a little more complicated. And that's randomly happens in all of the body. Means, even not only organ level, even in cell levels, those genes, which gene is activated, which gene is inactivated, they are differently regulated. And their pattern is also defined by methylation on the end. Another interesting example, so I hope to hear this basis. When I was young, the grandma said, hey, your bone is a character, your bone character is inherited from your father. Your skin is inherited to your mother. You have a better skin, and that's due to your mother. Blame your mother. Of course, that's my father's side of the grandma. At the moment, I said, hey, she didn't learn the Darwinian the genetics or the evolution. She doesn't know about the DNA. DNA should be mixed evenly and then transferred. Half and half should be transferred to the next generation. That's what we believe. That's the Darwinian evolution of the mechanism. But seems like this part, there is some of the more strange thing ongoing, so-called genomic imprinting. I don't know those pictures. So when they make this sperm and then egg cells, some genes, it doesn't matter whether it came from father or mother. Sperm side, it makes one side, it makes only father's genes methylation pattern follows. The other side, only mother's gene methylation pattern transfer. I said during those development, 99% are erased, but left over 0.1% of those genes. Some of them is that genetic information only follows those father side, father to son. It can be given to the daughter too, but that daughter's egg cell didn't know it. That's why that follows only to the father side, male side. Some other genes only follows those female side. And that regulation or that information, not genetic sequence changes. That's also methylation pattern. And very interestingly, those genes function is competition between male and female. Male genes tells, okay, when you make a baby, just the baby bigger and bigger and healthier and healthier. Take all the nutrients from mother. I don't care my partner, I care my baby. Because my baby carries my gene. So that's why even though sacrificing mother, baby's the survival, baby's health is more important to the male. In other terms, for mother, okay, this is my baby. It's important to me. But at the same time, I may have another chance to get another baby. I cannot fully sacrifice myself only for this baby. I tried to distribute those resources to different babies evenly. That is better for me. I have 2 babies now. I can have 2 more babies later. Right? So 2 babies, that's one done. 2 more babies is the double, right? There's a half information. So that kind of a gene competition is clearly shown here. So that's why if that gene is over the stress, if you combine those genes together, then babies is super big. If a mother side genes are combined together, babies are super small. So they are balance out each other. That's why the reasonable size of the babies can come. So that is very interesting topic, and also utilizing that concept of that's also through the DNA methylation, and that is one example of competition between female and male competition in terms of epigenetics. Okay. Why only in CPG? Well, methylation site left over is only C for now. If there's a U was in the DNA, it may be UPA could be chosen, but somehow in our nature, we chose this one as RNA DNA recognition. Only this one has been chosen. I would say this is almost random because we have the ATP, GTP, CTP, UTP in RNA, but out of 4 combinations for energy sources, what do we use? Mainly ATP. The energy is stored in triphosphate, right? It doesn't matter whether this is A or T or C or U. But we just, by chance, we chose A recognition and utilized this as the energy source. Maybe similar things. CPG was just chosen as one example and just set it down as a main switch. So that means maybe through this CPG area, there's a recognition of the enzyme, let's say HDAC. That is methylated, and HDAC comes in, recognize it, and remove the acetylgroup. Then that is the next step of closing the genes. Okay, so this week, the main topic, again, if you didn't have a chance, I would recommend, there's a lot more interesting studies about this epigenetics. If you read this book, then you can imagine the knowledge about it. Okay, now we are here. Next week, we'll talk about the stem cell and also how to make artificial stem cells, so-called IPS, induced pluripotent stem cells. And then we go into the middle term and some topics afterwards. So there's an open slot here. Still, we don't have volunteer for next week. So first come, first served. If you are interested, send me Kakao or email, but depending on which one I look at first, to be safe, you may send both. Okay, first one I see, then I will pick it up. Okay. Next example, let's just say making clones, like a Dolly study, would be interesting topic. It's one kind of utilizing stem cell. It's not really IPS, but that's the cloning of the whole body. And IPS can be another the interesting topic. We had some scam, unfortunately, the shameful, the scientific story in Korea, right? Utilizing stem cell or making stem cell kind of thing. So after that, there's another big scam in Japan too. Our case, and then Shina Yamanaka's successful IPS story, and another Obotaka, I forgot the lady's name. Japanese scientist problem of a big scam too. So maybe that can be another interesting story. So you can think about it, and then you can volunteer for next week the short talk. Okay. Any question for epigenetics for today? Okay, if not, see you next week. By the way, usually I open the next week of material. Now I come back to my office and usually last week I forgot it, maybe. If you see it's not still open, then most likely I forgot it, remind me. Okay, Professor, it's not open yet. What are you doing? Open now. Okay. Okay guys, see you next week.
교수님. 교수님. 교수님.